Hi, my name is Patrick Killian Jackson, and this is my final project for CS CM124, and I've chosen Project 1, which is on haplotype phasing. So, a haplotype is the sequence of nucleotides along a single chromosome, and it's often difficult to tell the difference between the two chromosomes in a pair, which leads to having a combined haplotype, which we also call a genotype. And so haplotype phasing, what that is a method used to discover what the haplotypes are when you're given a list of genotypes. So the motivation behind haplotype phasing, like why anyone would do it, is it provides a more complete view of an individual's genome. So haplotypes provide more information than genotypes do, so we can get just way more information out of the genetic data. And also, more motivation is haplotype phasing has many applications, and those include association studies, detecting positive selection, estimating recombination rate, understanding gene function, analysis of regions of the genome that are functionally related. My algorithm for achieving haplotype phasing uses an adaptation of Clark's method, which was discussed in the slides in class, to determine a list of haplotypes corresponding to a given set of genotypes. So next I'm going to kind of define the computational problem that we have. So the input to this problem is a text file that contains genotypes that have been translated into a form where the SNPs are translated to be either zeros, ones, or twos. And those correspond to 0 is homozygous reference, 1 is heterozygous, and 2 is homozygous alternate. And then the output of this algorithm is a text file that contains the haplotypes that we've derived from the input list of individual genotypes. And the goal that my algorithm has is for the list of haplotypes that we generate to have 2 haplotypes that correspond to each of the inputted genotypes in the input text file. And in this list of haplotypes, it won't necessarily be double the size of the genotype list because overlap is allowed since multiple genotypes may map to the same haplotypes. And then the benchmarks for this computational problem are computational time and the accuracy of the haplotypes that I compute. So the baseline method is based on Clark's method, as I discussed earlier in the presentation, and this is kind of a breakdown of how Clark's method works. So first, you scan through the genotypes and compile a list of haplotypes that we determine from the unambiguous genotypes. So for my testing purposes and for my algorithm, those unambiguous genotypes are the ones that consist of only zeros and only twos. And going back to the previous slide, those are homozygous reference and homozygous alternate. And then after compiling that initial list of haplotypes, we scan through the remaining ambiguous genotypes, and if 2 haplotypes in the building haplotype list can resolve an undetermined genotype, which means that that haplotype corresponds to the genotype where we can flip the zeros or ones if the original genotype started with a 1 and had a heterozygous. So for those, we can flip it and see which ones correspond to our building genotype list. So we can resolve an undetermined genotype. We don't do anything. We move on to the next genotype because we already have haplotypes that are in the haplotype list that can resolve that undetermined genotype. And then if one of the known haplotypes and combined with a new haplotype that's not in the haplotype list can resolve the genotype, then we add that new haplotype to the list. And so iteratively going through this, we can find a list of haplotypes that correspond to the inputted genotypes. And this is just a more visual breakdown of how Clark's method works. You see we start off with a list of genotypes on the left, and then in the first round we add to the known haplotypes the the ones that we can resolve since they're unambiguous, and then with each subsequent round, we compare the remaining ambiguous genotypes to the list of known haplotypes, and we can iteratively through each round figure out what the remaining haplotypes are. So I I didn't follow Clark's method exactly, but I have like an adaptation of it that I used for my algorithm for this project. And my adaptation starts by reading in the input file and storing all the genotypes in the input file in a global variable, which I call genotypes. And then after that, I scan through genotypes and compile a list in a new global variable called haplotypes of the haplotypes that I can derive from the unambiguous genotypes in the list, just like in Clark's method. And additionally, I store the remaining ambiguous genotypes in another global variable called undetermined. So those are the ones that we haven't yet found haplotypes for yet. And then we scan through undetermined, and for each genotype in undetermined, we generate a list stored in a variable called buffer of all of the possible haplotypes for that genotype. So we go through, and wherever the original genotype was a 1, we kind of split the haplotype, or sorry, the genotype into 2 new and add a 0 to 1 and a 1 to the other one, and we continue to split the list and build it until we have a list of all of the possible haplotypes that genotype could be. And then we compare each item in buffer that we just generated to our current list of haplotypes, and if there's a match, we add the corresponding haplotype, like the flipped version of the match, to the building list of haplotypes. And so after we've scanned through undetermined, we output all the members of haplotypes to an output text file, and that is the end of my algorithm. So a little bit of performance analysis. I ran, excuse me. I ran I used the easy test case as the basis of my tests. And so I started with just running the easy test case that was given in the data files. And then what I started to do was vary the number of SNPs, but keep the number of genotypes constant. So the file started with 60 genotypes, each who had 30 SNPs. And I went as low as 10 SNPs and as high as 39 SNPs. And if you look at this algorithm runtime graph here on this slide, I like times each time I ran the algorithm, I timed the runtime, and you can see that as the number of SNPs increases, at first it is a pretty gradual increase in runtime, but as the number of SNPs like really gets high, like you can see at 39, it took before previously it was under 10 seconds, and when you have 39 SNPs, it gets to be over a minute. So there's definitely an exponential increase for my algorithm in the runtime as the number of SNPs increases. Some additional performance analysis. So just considering the 2 input text files that were given to us in the data files. The first one I ran was the very easy training genotypes.txt, and doing that I observed my results in the output text file and compared them to the very easy training haplotypes.txt file, and saw that I generated the same haplotypes. So it was very accurate for the very easy training. And then I also ran the easy training genotypes.txt file, and it generated an output file containing the haplotypes matching those in easy training haplotypes.txt. However, there was one additional haplotype, so there was 15 in the example file, and I generated 16, and 15 of mine matched exactly with the 15 that were in the example file, but I had one extra haplotype, and I believe the reason for this is when I'm scanning through the list of ambiguous genotypes, I don't keep track of how many haplotypes I've added per genotype, and so there's the potential that it's not a completely minimal algorithm, and there's the potential of adding extra haplotypes in and finding more haplotypes. So it just scans through one time and it finds however many haplotypes that it can find. So just some thoughts just after doing this project and considering how the performance and the accuracy played out. There are several future improvements that I would make to my algorithm. First of all, unlike Clark's method, my algorithm, as I was just saying, only passes through the ambiguous genotypes once as opposed to doing it iteratively. And this is sufficient for genotypes with a limited number of SNPs. For example, the easy case with 30 SNPs. But as the data sets get larger and there's way more SNPs and way more genotypes, it definitely will be required to do it iteratively so that you don't just pass through one time, because you may not find all of the haplotypes in the first pass, and the haplotypes that you do find in the first pass will be helpful on the next pass to find even more haplotypes. So that's one feature improvement to my algorithm. Also, I think that another strategy could be way more efficient to avoid the overhead of generating all of the possible haplotypes, because I added a print statement in to see the length of buffer back here in the explanation of my algorithm. When I scan through undetermined, I generate a list of all the possible haplotypes for each genotype, and I noticed that there's a significant overhead here because it sometimes generates as many as 200,000 possible haplotypes for each genotype. So as the number of SNPs gets even larger, the number of possible haplotypes for each genotype just gets bigger and bigger and bigger. So this is definitely not the most efficient way to go about it. It was sufficient for my purposes, but definitely in the future I think that it could be improved by adding a more efficient way of comparing the haplotypes to the ambiguous genotypes. And then the last thing is I would somehow figure out a way to incorporate independence from unambiguous genotypes. So one thing with using Clark's method, or my implementation of Clark's method, is it is dependent on there being unambiguous genotypes in the input text file. So if you can't generate an initial list of haplotypes from the inputted text file, you're out of luck for my algorithm. So I'm not sure how to get around that, but that would be a huge improvement to my algorithm to not have to depend on there being unambiguous genotypes at the beginning. That is my presentation. Thank you very much.